Good day, dear students. Today we start the lectures on spatial histology, and the subject of today's lecture is the sensory system. And today we will talk about the organs of sense. You know that the central nervous system controls the functioning of our organs via receiving of information from them, from external and internal environment, and the perceiving information from external and internal environment of the body. It is a functioning of organs of sense. The term sensory organ, it means the spatial organ which recognize some exact irritation from outside or inside of the body. We know that in the human body we have two kinds of nerve endings. These are sensory nerve endings and motor nerve endings. Sensory nerve endings, they perceive information, various stimuli, and then here the information will be transformed into the nerve impulse, and the nerve impulse will be transmitted to the central nervous system via the nerves. In CNS, the information is analyzed, integrated, and the response of the CNS will be transmitted to the effector organ via the motor nerves, and here we are looking at the motor nerve endings. So today's subject is the sensory nerve endings, and today we will talk about their classification and their types. Classification of receptors there are several classifications. First of all, we have to understand that we have general sensations and spatial sensations. The general sense, it includes receptors that distributed throughout the skin and the organs. These are somatic receptors that located in skin and muscle, tendons. Then we have visceral receptors. They are located in internal organs. Spatial sense, it includes receptors that form the specialized organs. So that will be specialized organs which are differentiated to receive particular stimuli. For example, we have organ of vision that is specialized for receiving of light sensations. We have taste organ for perceiving taste of food. Then we have organ of smell or olfactory organ. We have organ of hearing, balance. So each of them are specialized for perceiving some particular stimuli. They compose our spatial senses. Then we have three criteria used to describe receptors. That is stimulus origin, receptor distribution, and modality of stimulus. Based on stimulus location, there are three types of receptors. They are extrareceptors, intrareceptors, and proprioceptors. Now we will look at the extrareceptors. What are they? The extrareceptors located near the body surfaces. It is the skin and mainly on the surface of the body, and they respond to the stimuli which received from the external environment. Intrareceptors, they located inside of our internal organs, and then they sense the position of our internal organs to providing this information for the CNS. Then proprioceptors. Proprioceptors, they are located in muscles, in joint capsules, in tendons, and fascias, ligaments, and they provide the CNS information about the position of the muscles and joints. Then this information goes to the spinal cord, and that will be unconscious information about the position of the muscles. Then from the spinal cord, this information will go to the cerebellum and the brain cortex that should be aware of position of our muscles to provide their control. Then we have to know that sensory organs are classified according to the receptor, to the nature of the receptor that receives the information. First of all, we have to know that primary sentient organs, what are they? They are receptor here in the primary sentient organs, sentient organs. The receptors, these are neurons, and actually the dendrites of neurons. As we know, in neurons, in nerve cells, the part of the cell which receives information, that is a dendrite. And so in these primary sentient organs, the information will be perceived by the dendrite of the sensory neuron. For example, it's in the case of the eye. In eye, the light stimuli will excitate the dendrites of the photosensory neurons. And so the eye belongs to the primary sentient organ. Then we have smell organ or olfactory organ, and here also the information, the sensory input will be perceived by the dendrites of olfactory neurons. So in both cases, the receptor, it is the peripheral part of the neuron. In secondary sentient organs, the receptors are epithelial cells. It means that the primary components which will perceive that information, it will be the epithelial cell. For example, in case of the organ of hearing, or organ of equilibrium, or the organ of taste. In this case, we have specialized epithelial cells via which the organ will take information, then it will go to the neuron. So that they are the secondary sentient organs. By that way, we can classify the spatial senses into three main groups. They are primary sensory organs or sentient organs. As we know, they are neurosensory, smell and visual organ. Then we have secondary sensory organs. They are taste organ and audio vestibular organ, or organ of hearing, and organ of balance. And third one, these are sensory endings or sensory peripheral nerve terminals. These are various types of receptors that scattered throughout our body, and they do not form spatial sensory organ. They are just receptors. Here we are looking at all of these kinds of sensory organs, and each of them, they have its own function. A little about the peripheral receptors, which we need to perceive various stimuli from the organs. The peripheral receptors, according to the type of stimuli that excite these nerve endings, they are classified into mechanoreceptors, thermoreceptors, and nociceptors or plurireceptors. The mechanoreceptors, according to the type of stimuli, they may be the touch receptors, receptors that receive the perceived vibration, pressure, and other kinds of mechanical stimuli. Here we are looking at some of them, for example. Then these are mechanoreceptors. Then we can see here thermoreceptors that will respond to the extremes of temperature, that may be if the temperature more than 43 degrees by Celsius or less than 30 degrees. Then we have nociceptors that will respond to extremes in mechanical stimuli, or extremes in temperature, or to the certain chemical substances like serotonin, bradykinin, and so they are pain receptors. Then, according to their structure, the nerve endings may be classified as free nerve endings. They lack any connective tissue components and the Schwann cells. They are, for example, peritracheal nerve endings. Peritracheal nerve endings we can find in the epidermis of the skin, mainly in the more sensitive areas, that is the face or lips or the cornea of the eye. There we can find the free nerve endings that located between the epithelial cells. And then we have another kind of these free nerve endings, that is Merkel cell complex. Merkel complex, it means that there is a Merkel cell, that cell of the epidermis that resides in the basal layers of the epidermis, and to its base comes an unmyelinated nerve fiber that form contact with this cell. On the surface of these cells we have microvilli, so these are to get the Merkel cell together with this nerve ending, they form Merkel complex. And the main function of this complex is perceiving of touch stimuli. It means that they are receptors for touch discrimination. Then we have another kind of the receptors. They are capsulated nerve endings. They have not just only Schwann cell coat, but they also have some connective tissue capsule. That's why they call capsulated nerve endings. Here we can see some of them. For example, Meissner's corpuscles, that is this one here. Meissner corpuscle, it is the receptor which we can find in eyelids, in face, also in skin of them, in the lips, and in nipples and other areas of the body which are very sensitive, also in palms. Meissner corpuscle, it includes several unmyelinated nerve fibers which enter into this corpuscle, and between these, each of these nerve fiber covered by Schwann cell. Between the nerve fibers inside of the Meissner's corpuscles, we can find the fibroblast-like cells or modified Schwann cells. They are located between nerve terminals. And all of the corpuscle is surrounded by connective tissue capsule. So these are also receptors for touch discrimination, and they are mainly receptors for fine touch, and they are specialized in perceiving the edges and points of the objects. Then we have here the Pacinian's corpuscles. Here in cross section they resemble the onion, and inside of the Pacinian corpuscles we can find one nerve terminal that run through all of the entire length of the corpuscle, as we can see here. And then this nerve ending is surrounded by several layers of fibroblast-like cells. Between each of these cells located fluid filled spaces. There are approximately 60 layers, concentric layers of fibroblasts. Around this core, we have additional layer of 30 fibroblast-like cells. And around all of this corpuscle, we have connective tissue capsule. And these are receptors which located in more deep layers of the skin, for example in hypodermis. Also we can find in mesentery and in joint capsules. They are receptors for vibration and pressure. During applying the pressure on the surface, for example on the skin, these cells they will press against each other, and the fluid between them will be pressed out. By that way this force of pressure will be transmitted to the nerve terminal, and nerve terminal will excitate, and here will take place depolarization, and the nerve impulse will go to the central nervous system. Then here we are looking at the Ruffinius corpus, Ruffinius endings. They are also encapsulated, and they located also in dermis. We can find them. Their capsule that insert into the collagen fibers of the connective tissue, and it makes them very sensitive for stretch. So they are receptors for the stretch receptors. Then we are going to the next. Here we can see the same receptors. This is the Meissner's corpuscles. These are peritracheal nerve endings, which are free nerve endings, as we told. That is the Ruffinius nerve endings. Then peritracheal and Krause's and bulbs. They are called receptors, but their exact function is the subject of further discussions. That is Pacinian's corpuscles. These are the receptors for the pressure. Now let's start talking about the eye, and initially we have to know that the eye, these eyes, they are receptors for vision, and they are located in hollow bony orbit. They have diameter approximately 24 millimeters, and they have some tunics. You know that each of the eyeball is surrounded by 3 tunics. They are tunica fibrosa, vasculosa, and neural tunic. Here we are looking how the eyeballs develop. Actually, the development of the eyeballs start at the 22nd week of gestation. The eyeballs will develop from 3 sources. First of all, that is the surface ectoderm. Then the optic vesicle, which is outgrowth of the diencephalon. Then we have mesenchyme. You know that the mesenchyme is embryonic connective tissue, which may originate from mesoderm or ectoderm, exactly from the neural crest cells. And the mesenchyme of the head and neck region mainly, mostly it takes its origin from the neural crest cells. So here we are looking how the optic vesicle, that is outgrowth of the neuroectoderm of the diencephalon, they are paired organs, which these are optic vesicles. They outgrow from the diencephalon and grow toward the in anteriorward direction. They will come towards the surface ectoderm. Here, at the attachment site between the optic vesicle and the surface ectoderm, they will mutually interact and mutually stimulate each other. As a result, this optic vesicle will undergo invagination, and invagination will deepen. Finally, there will form optic cup. The optic cap will include 2 layers: outer layer or outer epithelium and inner layer. And from the outer layer will develop the pigmented epithelium of the retina, and from the inner layer will develop the neural retina. So the layers of the retina will develop from the walls of the optic cap. Here we will have interretinal space that will further obliterate. The stalk, which will connect the optic vesicle and further the optic cup with the diencephalon, that stalk, optic stalk, further will develop into the optic nerve. Here we also can see that ectoderm, surface ectoderm, under the influence of this neuroectoderm that comes from the that comes from the neural tube. It will undergo also invagination, and here we will see the lens vesicle. Then the lens vesicle will detach from the surface ectoderm, and it will actually here first of all will be develop the lens placode. Then from the lens placode will develop the lens of the eye. The remaining part of the ectoderm will be recovered, and it will give rise to the corneal epithelium. So the epithelium of the cornea, it also originates from the ectoderm germ layer. So ectoderm germ layer will give rise to the lens and to the corneal epithelium. And under the actually here on the inferior surface of the optic cup, we also will have actually the invagination that we'll call choroid fissure. Into this fissure will enter mesenchyme, and inside of this fissure will develop hyaloid artery. Hyaloid artery will connect the will supply the lens, because lens devoid of blood vessels, so inside of the eyeball we will have hyaloid artery. Also the mesenchyme that enters into the optic cup lumen, it will give rise to the vitreous body. And so the further development, the distal part of the hyaloid artery will degenerate, but its proximal portion will give rise to central vessels of the retina, that will be central artery and central vein of the retina that will supply the innermost layers of the retina. So all of the other structures, including the corneal stroma, corneal endothelium, then all of the parts of the choroid, and also the bulk of the ciliary bodies and iris originate from the mesenchyme. And those two components, the ciliary body and iris, will be covered by the pigmented epithelium of the retina that will continue and cover the posterior surface. So that's how our eyes develop. Then let's go to the structure of the eyeball. Here we are looking at the anatomical appearance of the eyeball, and just I mentioned that the eye includes three layers, three tunics that cover them. First of all, that is tunica fibrosa. Tunica fibrosa, it includes two components. First of all, cornea, that's like watch glass. It forms the anterior-most portion of the eye. It covers just 1/6 of the entire fibrous tunic. The posterior most part, 5/6 of the fibrous tunic, is covered by the sclera. Sclera, it is the dense, irregular connective tissue. There are a little amount of the blood vessels, but mainly sclera composed of the first type of collagen, and between the fibers we can find fibroblast cells. Into the sclera insert the extraocular muscles of the eye, which will move eye inside of the orbit. Also from the external surface, the sclera is covered by the Tenon's capsule, and between the Tenon's capsule and the sclera we have episclera. It is a fat tissue. Then we are coming to the cornea. Cornea, it is transparent if the sclera is opaque. Cornea is transparent because it belongs to dioptric apparatus of the eyeball. The dioptric apparatus or dioptric system of the eyeball, it includes cornea, then anterior chamber, posterior chamber, then the lens and the vitreous body. They are the medias through which the light rays should enter the eyeball and reach the nerve cells of the retina, so they should be transparent. And so the cornea, it's transparent. It has 5 layers, as you can see here: the corneal epithelium, Bowman's membrane, a stroma of the cornea, predecessors layer, descendants membrane, and finally number 5, that is the corneal endothelium. It is simple squamous epithelium. Corneal epithelium, this one, it is stratified squamous non-keratinized connective tissue, non-keratinized epithelium, and it is characterized by high turnover of cells. They regenerate during 7 days because in basal layers we have stem cells for it. Then the Bowman's membranes membranous structure, which maintain the epithelium and prevent entrance of foreign substances into the stroma. Stroma is connective tissue. It includes collagen fibers, which form here lamelles. Approximately 200 to 250 lamelles we have here. They located on the top of each other, and between these lamelles we have a lot of fibroblast cells. Then the ground substance include keratan sulfate, dermatan sulfate, these are proteoglycans, glycosaminoglycans. Then and the predecessors membrane and descendants membrane, they also form support. And finally the endothelium. Actually, we have to know that the cornea is avascular. It means that there are not any blood vessels, and their nourishment is provided by the aqueous humor, that is the fluid which fills the anterior chamber and posterior chamber of the eye. And between the cornea and the sclera there is the limbus, the region that is very important for drainage of the fluid from the anterior chamber. Here in this region, that is iridocorneal and iridocorneal angle. At this region, the collagen fibers of the cornea, they separate from each other, and between them there are formed spaces. So that is trabecular meshwork, and this is the outflow way for the fluid aqueous humor, which will then flow into the Schlemm's channel. Schlemm's channel is present between the cornea and sclera, and this channel has connection with the venous system of the eyeball. So the fluid will go out from this region. Then that was the fibrous tunic. Now let's come to the next tunic of the eyeball, that is the vascular tunic of the eyeball. It includes 3 parts. They are the choroid, then the ciliary body, and the iris. The choroid, if we will look at the choroid, we can see that it is the loose connective tissue. It reach in the blood vessels. It reach in the pigmented cells. And here we can distinguish some sub-layers inside of the choroid. So here we are looking at the sclera, then the choroid, and the retina. These are layers in the posterior wall of the eyeball. Here we can distinguish the suprachoroid lamina, then we have choriocapillary lamina, and finally we have here the Bruch membrane. Bruch's membrane, it is a membranous structure which includes elastic fibers and the collagen fibers, and that is the membranous structure which is interposed between the basal lamina of pigmented epithelium of the retina and the basal lamina of the blood capillaries, which present in choriocapillary layer. Here there are a lot of blood capillaries which participate in the blood supply of the retina. Then here we can see all of the components of the eyeball, and what we should pay our attention here. Pay attention to the choroid here, and this choroid which continue into the ciliary bodies, and here we can see the iris, which is the diaphragm with the central aperture that is a pupil. And here we also can see the extrinsic muscles which insert into the sclera, and pay attention to the central artery and central vein, which are actually in this, we can see them in the center of the optic nerve that enter the eyeball through the disc of optic nerve at the region of the disc of optic nerve, and the artery will supply inner layers of the retina. Pay attention to the iris here, and where the iris continue into the ciliary body. Ciliary body, it is a dilated portion of the vascular tunic, and here we can also see the sclera and the continuation of it into the cornea. So the canal of Schlemm we can distinguish here. That is the trabecular meshwork through which the fluid, the aqueous humor, will enter into the canal of Schlemm, and which will provide drainage of the aqueous humor. Pay attention to the layers again in the wall of the eyeball, posterior wall of the eyeball. Here we can distinguish sclera, then the choroid, and the retina. So choroid is, as we just mentioned, there is choroid capillary lamina and the Bruch membrane. Just mentioned that between the choroid and the retina is interposed the Bruch's membrane. Here we are looking at the histological sections of the choroid, at the different parts of the choroid. Here we can distinguish the blood vessels inside of this tunic of the eyeball and the pigmented cells. Okay, now we are coming to the ciliary body. Here we are looking at it, and the iris also. Both of them covered by the continuation of the pigmented epithelium of the retina. And we can see that on the surface of the ciliary body there are a lot of ciliary processes, and from the ciliary processes arise fibers. These are collagen fibers which form the ligamentus suspensory of lens, because they will insert into the lens capsule. They are fibers of zonula. And on the surface of ciliary processes, the pigmented epithelium here is lined by the one layer of columnar shaped cells which devoid of pigment. These are the cells which participate in the formation of the aqueous humor. So because the ciliary body and ciliary processes, they reach in the blood capillaries, the plasma of blood escape from there, from the capillaries, and from the plasma of blood these columnar shaped cells produce that fluid, and the fluid enters the posterior chamber. Then that is the iris here, that is the iridocorneal angel, and that is a cornea. So the fluid from posterior chamber of the eyeball will flow into the anterior chamber via the pupillary aperture, and then we know that at this angel we have trabecular meshwork that lead to the Schlemm's channel, and here we can see also that is the iris, the cornea, and the sclera. And Schlemm's canal again we can see here. So that is how the fluid produced and where it will flow out. If there is, in the case of problems in production of the fluid and the outflow ways, if there is some disturbances in the outflow also, the fluid aqueous humor is accumulated inside of the eyeball, and it leads to increase of intraocular pressure that leads to glaucoma. That is a pathology which, if it left untreated, it may lead even to blindness. Then the iris. Iris is the third part of the vascular tunic. It also reach in the pigmented cells, also blood capillaries. It composed of the loose connective tissue. Anterior surface of it also covered by endothelium. That it is the continuation of the endothelium that covers the posterior surface of the cornea. And then actually the anterior surface of the iris, it's truncated, there is a lot of fissure, it is irregular, and on the whole of it we have the loose connective tissue which form two border membranes. It will be anterior and posterior here, the limiting parts. Then the blood capillaries, and here we should know that under the pigmented epithelium, here we have a lot of cells which have radial orientation, and they form the muscular dilatator of pupil, and so that will be the muscle that will dilate pupil, and it is under the control of the sympathetic nervous system. That is a division of the autonomic nervous system. But around the pupil we have another group of muscles. They form sphincter pupilla muscle, that is under the control of the parasympathetic division of the autonomic nervous system. Here we are looking at this histological section of the iris, and in the posterior surface we can distinguish the pigmented epithelium here, which is the just here we looked, and under the dilatator pupilla muscle, which is located just under, then sphincter pupilla muscle. And as we mentioned, there are a lot of pigmented cells, so the pigment of the iris will impart the color to our eyes. So the color of our eyes will depend on the pigment, amount of pigment in the iris. These pigmented cells will prevent passage of light rays through the iris unless the pupillar aperture, so just through the aperture may light rays pass, so the iris prevent free passage of the light rays. Here we also can see the sections, histological sections of the posterior wall of the eyeball. For example, this one here, that is a retina, choroid, the angel here, that is the iris, and ciliary processes, also lens. Ciliary zonule, that are the fibers that come between, they run between the lens capsule and the ciliary processes. Actually, the ciliary body, we have to know that it include ciliary body include the muscle, ciliary muscle, that participate in the accommodation. It is the focusing of the light rays on the retina. That is actually the direct function of the lens. But the fibers, muscle fibers of the ciliary body, they will control the thickness of the lens. Actually, here we have the muscles which have in three-dimensional orientation, and during contraction of the ciliary muscles, the zonula fibers, they relax and the lens becomes thicker. By that way we can see nearby objects, and when the ciliary muscle relaxes, the zonular fibers they stretch, and it leads to decreasing the diameter of the lens, and by that way we can see far objects. So that is how the ciliary muscles and the zonal fibers and the lens will participate in the focusing of the light rays that is necessary for our better vision of the far and nearby objects. That ciliary processes which are covered by two layers of epithelium, the non-pigmented epithelium and pigmented epithelium, and these are those cells which produce that fluid. The lens, it is biconvex disc. It is transparent and it's avascular like the cornea, and it's also nourished by the fluid of the aqueous humor in the anterior and posterior chambers. And what can we see here? The under the epithelium, so that lens is covered by capsule. You know that lens is ectodermally derived structure of the eyeball, and that's why it's in its structure we can see the result or the source of its development. So under the capsule we can see the epithelium, it's anterior epithelium of the lens anterior epithelium. That is one layer of cuboidal shaped cells. The apexes of these cells, they faces the central part of the lens, and they gradually these lens cells become elongated, and they become hexagonal lens fibers. They lose the nuclei and move towards the center of the lens. Here they form nucleus of the lens, and that is the central core, which here the lens fibers devoid of nuclei, but they reach in the crystalline protein that provide transparency to the fibers. So that is the lens. Now we are coming to the last part before the retina. We are talking about that is a vitreous body. Vitreous body is gel-like substance. The 99% of it is the water, which is maintained here by the presence of hyaluronic acid, and also present here collagen fibers. All of those components are produced by hyalurocytes, which are present inside of the this vitreous body. And actually inside in the center of the vitreous body we can see hyaloid channel. It is a remnant of the hyaloid artery that in embryonic life it connected the posterior wall of the eyeball with the lens, and this artery carried blood, and it was necessary for the blood supply of the lens. But further in last trimester of the embryonic life, the hyaloid artery degenerates, and in its place we will see just hyaloid channel, and then it will remain as ligament. That is the vitreous body. It forms or it fills all of the internal part of the eyeball and provide its shape. Then the last tunic of the eyeball, as we know, that is the neural tunic or the retina, which include pigmented epithelium and the retina proper. As we know that retina, it developed from the neural tube. It's as evagination of the neural tube, so its structure will reflect its origin inside of the retina. So we will have two kinds of cells, as in other parts of the nerve tissue. There we will have glial cells and the neurons. And here we have to mention that the layers of the retina, the first layer here, that is pigmented epithelium, then the second one, the layer of rods and cones, it develops as the first layer in the wall of the neural retina, and from the mantle region of the mantle layer of the neural retina develop all of the neurons, cell bodies of neurons, and finally the marginal layer give rise to the nerve fibers. Here we will have nerve fibers that will go out from the eyeball, and they will form the optic nerve. Here that is the vitreous body. So the retina, it includes actually 10 layers, beginning from the pigmented epithelium until the layer of the till the inner limiting membrane. And here we have to talk about, first of all, Müller cells. The Müller cells, these are glial cells, which are supporting cells of the retina, and they create microenvironment for the proper functioning of neurons of the retina. Here we are looking at the chain of three kinds of neurons that join each other, and finally they serve for conveying of information, visual information, to the brain. The first neurons here, these are photosensory neurons. These are the second one, associated neurons, and finally ganglionic neurons. The photosensory neurons, actually all of them, they are the neurons, and we know that each neuron has the cell body, dendrite, and the axon. And here we also can see this order, and also we have to remember that the nerve cells receive information via the dendrites, then they carry this information to the cell body, and via the axon they send information to nerve impulse to the neighbor cell. And this law, dynamic law, we can proceed here also. So that are pigmented epithelium, and pigmented epithelium, it includes one layer of polygonal shaped cells. They join each other via tight junctions. There are also desmosomes between them and the gap junctions. By the formation of the zonal occludents, they form here blood-retinal barrier that are important in the isolation of the nerve cells from the blood-borne substances. Then pigmented epithelium also, because it includes melanin granules, it prevents reflection of the light rays from the posterior wall of the eyeball. So by that way it prevents distraction of the light. So the other function of the pigmented epithelium, its active role in the photoreception. As we know, the pigmented epithelium, because they store vitamin A derivatives, it participates in the esterification of its derivatives, and it plays active role in photoreception. Also, the pigmented epithelial cells, they surround the tips of the rods and cones, and they phagocytose their spent membranes. By that way, they help in regeneration of the membranes in rods and cones. And now we are looking at the photoreceptor cells, and here these are the associative neurons of the retina and the ganglionic cells. So as we see here, the photoreceptor cells, they have nuclear region, then dendrites and axons. Actually, as we see here, there are two types of photoreceptor cells. These are rods and the cones. Each of them has the same parts. Actually, the rods, they are more in number, approximately 120 millions of rods we have in our retina, and approximately 3 to 4 millions of cones. The rods, their peripheral part include, so the dendrite include two parts, that is outer segment and the inner segment. The outer segment, it includes a stack of lamella which located on the top of each other. Each of the lamella include membranes that contain photopigment, rhodopsin photopigment. The rhodopsin, it is a complex of transmembrane protein of opsin and retinal, it is aldehyde of the vitamin A. Then in inner segment, we have organelles. These are endoplasmic reticulum, Golgi apparatus, mitochondria, and also between the outer and inner segment we have cilia which connect them. At the base of the cilia there is a basal body. Then the nuclear region we can see here, and the axons. Axons of photoreceptors, they form synaptic connections with the dendrites and processes of associated neurons. Their synaptic contacts is taking place in the layer which is called plexiform layer, that is the outer plexiform layer. The cones, these are, if the rods are receptors for dim light, and they produce nerve impulse from the single photon. Then the cones, these are receptors for color vision and for the bright light. The peripheral parts, the outer segment of the cone has shape more of the conical shape, that's why its name. And the outer segment of the rods, of the cones, they contain not separated discs, membranous discs, but in this case, in case of the cones, the membranes after invagination, they do not detach from each other. That's why all of the lamellas, they are interconnected with each other at the most peripheral part of the outer segment. Then also, as we know, the cones, these are receptors for bright light and color vision, and their receptors, as we see here, that is the structure of the cone, and that is the rod. Here we are looking at the cilia which connect the outer segment with the inner segment. In the cones we have the photoreceptor which is called iodopsin or photopsin. It's also a complex of opsin and retinal, but that is another isomer. And also here we should know that in the case of the cones, we have three kinds of the photopigment, photopsin, that is, each of them is sensible for certain wavelengths of light. So we have medium sized, we have long wavelengths and short wavelengths photopsins, and accordingly we have three kinds of cones, each of which contain this type of the photopsin pigment. We can see other shadows of light because of the excitation of various amounts of these cone types. And the inner, the nuclear region and the synaptic region of the cones, it has also the similarity with the synaptic region of the rods. Now we are going to the mechanism of photoreception, how we will see visual acuity and how it will take place. For that reason, we know that the way how the nerve impulse will generate it and how it will be conducted. Before talking about it, we have to mention about, talk about the glial cells of the retina, which are Müller cells. These are the cells which span all of the thickness of the retinal layers, and these are actually kinds of astrocytes. Inside of the Müller cells, the Müller cells will provide physical nutritive support for the retina, and these cells will create microenvironment for the proper functioning of the neurons of the retina. They provide also an insulation of the cells, and there are also records for their participants in photoreception process. But main function of them, it is supporting neurons, and also they clean intercellular space from the excess of neurotransmitters and the ions, especially potassium ions, which are released from the neurons during the photoreception. So these are the Müller cells, and the peripheral parts form outer limiting membrane, and inner processes form inner limiting membrane of the retina. Then we have here associative neurons. Associative neurons are of three kinds that are bipolar cells, which have one dendrite, one axon. These are bipolar cells. They form connections between the photoreceptors and ganglionic cells. And we have also horizontal cells and amacrine cells. Both of these kinds, they serve for lateral inhibition. It means that they provide passage of nerve impulses just from the photoreceptors, which are excited with the light, and they prevent passage of nerve impulses from the neighbor cells. So the horizontal cells, they will inhibit information at the level of photoreceptors because they form synaptic connections with the photoreceptors. And here we are looking at the amacrine cells. Amacrine cells provide inhibition of synaptic connections between the bipolar neurons and the ganglionic cells. And so here we also have plexiform layers, the outer and inner plexiform layers. Here we can see the synaptic connections between the nerve cells of the retina. Here we have two kinds of synaptic connections. There are flat synapses, which are formed between two cells. In this case, for example, the axon of the bipolar cell and the dendrite of the ganglionic cell. But in for example, we have also invaginated synapses. In this case, will participate the axon of one, for example, bipolar cell, and two dendrites, which belong to either amacrine cell and ganglionic cell. So, or both dendrites, which come from the ganglionic cell. So these are invaginated synapses. About the ganglionic cells, ganglionar cells, they are less in amount than previous kinds of cells, and as we see, more photoreceptors. Then we have associative neurons, and less amount we have ganglionar cells, and all of these specializations are serving for summation of information that comes from the photoreceptor cells. The ganglionar cells we have of several kinds. These are W, X, Y cells. Also in the retina, in human retina, there is photoreceptor glial cells, which also present here. These are actually kinds of ganglionar cells, and they contain melanopsin pigment. Their main function is transmission information to the superior suprachiasmatic nucleus that will inform the pineal gland about the day or night period of the light or night or dark period of the day. You know that pineal glands, these are endocrine glands which participate. Pineal gland, it is a gland which controls the circadian processes of the human body. So the photosensitive glial cells of the retina will provide information for the pineal gland about light or dark periods of the day. So these are also types of ganglionar cells which we have in our retina. Then, okay, as we see here, the light which enters the retina, it first of all passes through all of its layers till it reaches the photoreceptors till it reaches the cones and rods. And here we are looking at the pigmented epithelium of the retina that absorbs most of the light that enters the eye wall, and it prevents the reflection of the light from the posterior wall of the eye wall. It has a very important role in photoreception. After entrance light, which it passes through all of the layers, it will impinge the rods and cones and will excite these receptors. During excitation of the receptors, the nerve impulse is formed and it will transduce toward the brain in the way that it is opposite to the way of the light that entered the retina. So as we see here, arrows indicate the way of nerve impulse that will pass from, first of all, from the photoreceptors to the associative neurons, then into the ganglionar cells, and the axons of ganglionar cells, which form the nerve fibers, they will exit from the eyeball at the region of the disc of optic nerve. Here all of the nerve fibers come together, congregate, and they exit the eyeball. By that way, the nerve impulse will go to the subcortical and cortical centers of vision. In the center of the optic nerve, we are looking at the central artery and central vein that enter the retina, that enter the eyeball, and this central artery supplies the inner layers of the retina. The outer layers of the retina are supplied by the blood vessels of. Now let's come to the mechanism of photoreception. Actually, we know that in the outer segment of rods and cones, the membranes of lamellas, they include the photopigment. It depends on the cell. If it is a rod, it contains rhodopsin, and in case of the cone, there are the photopsin pigment. And at the dark period, the rhodopsin, it is connected. Rhodopsin, it is the complex of retinal and opsin protein or scotopsin protein. And at the dark period of the day, in the outer segment we have sodium channels, which are kept in the open state by the cyclic GMP. So cyclic GMP binds to the channel and keep it open state. The sodium ions continuously enter through the outer segment into the membrane, into the cell, and in the inner segment we have the sodium potassium ATPase or pump that carries sodium out of the cell instead of in exchange to two potassium ions that enter the cell. So continuously sodium ions enter, and via the inner segment they are going out. Also in the inner segment we have potassium channels that carry potassium ions out from the cell. And that is so that process continues. That's why inside of the photopigment cells at the rest we have potential, resting potential, which is less than in other cells of the body. There is minus 40 millivolt. That is in the rest state. But when the light imprints when the light will imprint the rods, what we will see that rhodopsin, which is activated by the light, it will subdivide into opsin and retinal. Why it is to answer this question, we have to look at the structure of the 11 cis isomer, and that has bent structure, and under the influence of light it changes and it becomes all trans isomer or all trans retinal, which have straight structure. And this arrangement, it is not accessible for binding with the retinal. That's why with the opsin protein, that's why the rhodopsin will break down into the opsin and all transretinal. Let's look here, and here we are looking at the rhodopsin, which is include opsin protein, transmembrane protein, and 11 cis retinal. Under the influence of light, here we will see that 11 all transretinal is formed, and it detaches from the opsin protein, and the opsin becomes activated, and then it leads to activation of the transducin. It is G protein, it is second messenger. The transducin activation will activate phosphodiesterase enzyme. Phosphodiesterase enzyme in turn will break down cyclic GMP. As we know, cyclic GMP is needed to keep the open state of these channels, sodium channels, that present in the outer segment. So low amount of GMP and sufficient amount of cyclic GMP inside of the cytoplasm lead to closing the sodium channels. So the sodium ions cannot enter the outer segment, and that lead to hyperpolarization of the cell. If we will look here, so the sodium ions cannot enter, as we see here, because the channel is closed, but the sodium potassium pump operates continue to carry out sodium ions. So amount of sodium ions inside of the cytoplasm decreases, and it leads to decreasing of the potential from the minus 40, it becomes minus 80, minus 90 millivolt, and it is that condition we will call hyperpolarization. Hyperpolarization of the cell, as we see here, the same condition, the activation of the phosphodiesterase by the transducin or G protein leads to breaking down of cyclic GMP, and sodium channels are closing here, and sodium cannot enter the cell. That leads to hyperpolarization, and it will result in the release of the glutamate neurotransmitter from the nerve terminal, from the axonal terminals of the photoreceptor cells. Glutamate will activate the bipolar neuron, and bipolar neurons will transmit information to the ganglion cells. By that way, here we have to pay attention to the way of activation of the photoreceptor cells. That is unlike in other somatic cells of the human body. It is a result of not depolarization, but of the hyperpolarization of the cell that lead to release of neurotransmitter and to conducting nerve impulse to the brain about the light impingement and about the visual information to the brain cortex. And additional accessory apparatus of the eyeball, they are eyelids, then we have lacrimal apparatus, then we have muscles that move the eyeball in the orbit, and we have conjunctiva. So here we are looking at the histological section of the eyelids. Eyelids, you know that they also develop from the surface ectoderm that cover its surface, and inside there is a mesenchyme that will form its connective tissue elements, also the muscles. The external surface of the eyelids covered by very thin skin, and the dermis, it includes here the sebaceous glands, then sweat glands, and here the hair follicles we can find. The dermis here don't contain adipose cells. And inner surface of the eyelids, it is covered by conjunctiva, conjunctiva, it will also cover the anterior surface of the eyeball, exception the cornea. Region of the cornea is not covered by the conjunctiva, but surface of the sclera is covered by conjunctiva, and then the also sclera, then the eyelids. Histological structure of the conjunctiva we are looking here. It is covered by stratified columnar epithelium that contain also goblet cells that produce mucus, and it will be added to the tear film that cover the anterior surface of the eyeball, and it will lubricate and protect it from the dehydration. Then under the epithelium we have lamina propria, which is connective tissue layer of the conjunctiva, and then inside of the eyelids we have tarsal plates, which are plates of dense connective tissue, and then here we are looking at the muscles, striated muscles inside of the eyelids, and along the internal edge of the eyelids there are a lot of glands. These are especially meibomian glands, which are modified sebaceous glands, and they open onto the they open freely on the free edge of the eyelids. Here we are looking at the secretory portions of these glands. And on the free edge of the eyelid we have the 3 to 4 rows of eyelashes. Into the follicles, hair follicles of the eyelashes open glands of mole, which have a spiral way, and the ducts open into these. These are actually modified sweet glands. Then we have also glands of Zeiss, which also open into the eyelashes, into the ducts, into the follicles of the eyelashes. Then here we are looking at the meibomian glands. The conjunctiva we are looking here, that is a margin of the eyelid. Here the inner surface of it covered by the conjunctiva, and outer surface, that is the skin, and then the glands of the eyelids, and then lacrimal apparatus. Lacrimal apparatus include lacrimal glands, and they located in lacrimal fossa in superior lateral margin of the eyeball, and they are compound tubular alveolar glands, which here we are looking at the secretory portions of the lacrimal glands. They produce tear, which is produced via the ducts, approximately 10 to 12 ducts. They open into the conjunctival sac, and from the conjunctival sac, by the blinking, this tear will moisten anterior surface of the eyeball. Also inside of the lacrimal fluid we have lysozymantic bacterial substances that will protect the eye from the, not just from the hydration, but also from the any bacterial invasion. Then the lacrimal fluid is accumulated at the punctum, at the medial edge of each eyelid, and from there will start canaliculi, lacrimal canaliculi that lead to lacrimal sac, from which arises nasolacrimal duct to open into the nasal cavity. Okay, now we are coming to the olfactory organ. Olfactory epithelium is located in the superior, in the roof of the nasal cavity, in the superior concha surface of it, and also superior region of the internasal septum. Here we are looking at the olfactory epithelium. This epithelium includes 3 main kinds of cells. There are olfactory cells. These are yellow colored cells here. These are actually neurons, nerve cells. And as neurons, they have dendrites which go to the periphery. And here we can see the surface, that is the roof of the nasal cavity. that is covered by mucus, and so the dendrites of these olfactory cells near the surface of the olfactory epithelium, they form dilatation. That region is called olfactory vesicle. From it arise cilia, approximately 6 to 8 to 12 cilia arise from each of the olfactory vesicle. And on the surface of cilia we have receptors for odoriferous substances. The axons of the olfactory neurons, they pierce the first of all they will be covered by Schwann cells, and they will form non-myelinated nerve fibers. Then they will pierce the cribriform plate of ethmoid bone and ascend towards the brain. And on the inferior surface of the brain we have olfactory bulb that includes second neuron on the way. So first neuron, these are olfactory neurons, and second one, located in the olfactory bulb. They also called mitral cells. Mitral cells, via the dendrites, receive information from these unmyelinated nerve fibers, which form olfactory nerve. They also call filia olfactoria. And then this from the axons of these cells will form olfactory tracts. So that is the olfactory epithelium. Also between the these nerve cells we have supporting cells, which have microvilli on the apical surfaces, and there are also basal cells which serve as stem cells, and there are two kinds of them, horizontal and globose cells. The division of these cells will produce new neurons and new supporting cells. The rate of renewal here approximately 3 months, 3 to 4 months. So, and under the epithelium we have lamina propria of mucosa, of olfactory mucosa, that contain the glands, the olfactory glands. They also called glands of Bowman. Here we can see them, and this is the secretory portion, and that is a duct of this gland. So the secret which is produced in Bowman glands, they released onto the surface of olfactory epithelium to moisten it, and also inside of this fluid will be dissolved the odoriferous substances, and this mucus, that fluid also will concentrate these odoriferous substances near to the olfactory cells and will prevent dissipating of those substances from the olfactory epithelium. So that is the olfactory. And finally we are coming to the ear. That is we know that ear, it includes two kinds of the sensory organs. There is the organ of hearing and organ of balance. Both of these sensory organs are present inside of our ear. Anatomically we know that ear include external ear, which contain pinna or auricule and external auditory meatus. Then we have middle ear cavity, and we have inner ear. So the external auditory meatus, here it is auricle. It is it includes plates of elastic cartilage that covered by very thin skin. There are sebaceous glands, sweat glands. The external auditory meatus, it includes the cartilaginous portion and the bony portion. It is covered by skin, and inside of the skin we have hair follicles and also ceruminous glands, which are modified sweat glands that produce cerumen. It's waxy substance, and it also contains antibacterial chemicals which needed for the protection of the ear from any invasion, bacterial invasion. At the end of the external ear we have tympanic membrane. Tympanic membrane, it is the membranous structure which has three sources for development. It developed from the surface ectoderm that gave rise to its external epithelium, that is epidermis. Then inner epithelium of the tympanic membrane it has endodermal origin, And the middle part, the connective tissue elements of the bulk of the tympanic membrane, it has mesodermal origin. Middle ear cavity. Middle ear cavity, it is a tympanic cavity, that air-filled cavity. It is also covered by epithelium. This is simple squamous to simple cuboidal, and also it has connection with the oropharynx via the it has connection with the nasopharynx via the eustachian or auditory tubes. And actually we know that middle ear cavity, it developed from the first pharyngeal pouch, and it has endodermal origin. It is covered by pseudostratified ciliated epithelium, which is respiratory epithelium actually. And the entrance of this tube also covered by pseudostratified epithelium. Under of it we have lamina propria, that is connective tissue elements. And the roof of the tympanic cavity, and most part of it covered by simple squamous to cuboidal epithelium that adheres to the underlying bone. That's why there are no lamina propria. That's why that regions are more susceptible for infections. In the middle ear cavity, we have three bony ossicles, malleus, incus, and stapes, and they are joined to each other, articulated in very movable joints. And the malleus is attached to the tympanic membrane, and the stapes attached to the oval window. And the oval window, together with the round window, they are connections between the middle ear cavity and the inner ear cavity. Then there are two muscles, the musculotensor tympani, and these muscles necessary for maintaining, for protection of the tympanic membrane and the oval window membrane from the damaging effect of the loud voices. They lead to maximal tension of these membranes and prevent the damage by very loud voices. Eustachian tube, it provides equalization of the air pressure inside of the middle ear cavity with the air pressure outside of the body. And then the inner ear. Inner ear, it is a labyrinth. It's composed of the bony labyrinth, which contains the membranous labyrinth. The inner ear, it includes bony labyrinth. Here, it actually developed inner ear, it developed from the otic vesicles, and this otic vesicle then in embryonic life it subdivides into two parts. From its anterior portion develops the cochlea and the sulculus, and from the posterior portion develop the utriculus with the semicircular canals, and then they form connections between each other. In the bony labyrinth we can distinguish the bony cochlea, and then vestibulum, then semicircular canals. There are three semicircular canals. One end of each form dilatation, that is ampullar, and the two ends of the channels, they fused with each other. That's why into the vestibulum open 5 openings instead of 6. The bony labyrinth, it filled with a fluid that is a perilymph. Inside of the bony labyrinth we have membranous labyrinth, which have membranous walls, and so accordingly inside of the cochlea we have the spiral, or inside of the bony cochlea we have membranous portion, membranous labyrinth. Actually the cochlea, as we know, it is formed by the spiral canal, which form 2.5 turn turns around the central axis, that is a modiolus. And that's why inside of the cochlea, bony cochlea, inside of the cochlear duct, cochlear canal, we have cochlear duct, as we can see here. It also forms to a half turn around the central axis. In the wall of the cochlear duct, we have the organ of Corti. It is the organ of hearing. In the vestibulum, we have two sacs, that is the sacculus and utriculus, which are interconnected with each other via the ductulus utriculosacularis. And from a short end of each of them, there arise canal that fuses with each other, two canals, and they form endolymphatic duct that terminates at the endolymphatic sac. Between the sacculus and the cochlear duct, we have ductus reuniens. Then, inside of the semicircular canals, we have semicircular ducts. And so all of the membranous labyrinth is filled with a fluid, which is the endolymph. Endolymph, its composition is near to the composition of the intracellular fluid, and the composition of the perilymph, it is near to the extracellular fluid. So these are the parts of the membranous labyrinth. And as we know, in the wall, along the wall of the spiral duct, there is the organ of Corti, or organ of hearing. And in the wall of the sacculus and the utriculus, we have specialized receptor areas, which called maculae. Respectively, we have a macula of the sacculus, which is located in its wall, and macula of the utricule, which is located in its bottom. That's why they are actually receptors for organ of balance and or equilibrium, and they are responsible for linear accelerations of the body and for the gravity. In the inside of the semicircular canals, and exactly in the ampullar regions, we have also specialized receptor areas, which called cristae. They also belong to organ of equilibrium and balance, but those organs respond to angular accelerations of the body. And so let's go to the here we are looking at the histological section of the external auditory meatus, that we know that it's covered by epidermis, that is the skin. And here we also have hair follicles and ceruminous glands. Their ducts open onto the surface of the skin, and as we know, they produce waxy material. And the dermis, so these are the components of the external auditory meatus. Here we are looking at the cochlea, and from the cochlea arises a cochlear branch of the cranial nerve aids, and then here we are looking at the crista ampullaris, which located in the ampullar regions, and the vestibulum that contains sacculus and utriculus with their receptor areas. In cross section of the semicircular canal, we are looking at the semicircular duct, that is a bony canal that contains perilymph, and that is endolymph filled membranous canal, or that is we know that that is the semicircular duct. And in the cross section of the cochlea, we are looking at in the cross section of the cochlea, we are looking at the cochlear duct or spiral duct that include endolymph, and by location of the endolymph field spiral duct, all of the canal, this spiral canal lumen, subdivided into two parts, two portions. The uppermost is called scala vestibule and lowest is called tympani, scala tympani. That is the structure of the macula, which is in the wall of the utriculus and the sacculus. That is macula and macula of saccule and utricule respectively. Here we are looking at the receptor area, which contain receptor cells. Here we have two kinds of cells. These are type 1 and type 2 hair cells. On the apical surface there are stereocilia, and the basal parts of them, they form connections with the nerve terminals. These are actually dendrites of nerve cells which reside in vestibular ganglion. So first type of cells have rounded base and narrow apical portion, and this rounded base is surrounded by afferent nerve ending. And the second type of epithelial, neuroepithelial sensory cells, they have columnar shape, and on the base of them there is focal connections, synaptic connections. And then the cilia, the stereocilia, are embedded in gelatinous membrane, which is called otolithic membrane, on the surface of which there are the otoconia. These are otoliths. These are crystals of calcium carbonate, and they are located on the surface of this membrane. Because they have weight, they lead to changing, when the changing of the position of the head, they change position of the otolithic membrane, and it leads to excitation of the nerve terminals, and the nerve impulse is going to the brain via the vestibular portion of the vestibulocochlear nerve. And the structure of the, here we are looking at a histological section of the macula, and these are otoliths, that are otolithic membrane and hair cells. And between the hair cells we are looking at the supporting cells, which located between and under these hair cells. And the crista ampullaris. Crista ampullaris, they are located in the ampullar regions of the semicircular ducts, and in this region the bony wall form ridge, on the surface of which there are receptor cells, and that is the endolymphatic field space, that is the crista ampullaris in histological section, and that is perilymph field bony labyrinth. Then we are coming, and actually the structure of the crista ampullaris, it has similarities with the structure of the macula, but the difference here, it is in the structure of the membrane that is located over the hair cells. There are also hair cells first and second type, and supporting cells, but instead of otolithic membrane, on the surface of that epithelia is located cupula, which has cupular shape. It's also gelatinous mass. And the crista ampullaris, these are receptors for angular acceleration of the movements, and they will excite it when we turn our head in three-dimensional orientation, and it will lead to excitation of nerve endings. Here we are coming to the cochlea. As we know, cochlea includes spiral duct, which form also 2.5 turning around the bony modulus. Inside of the bony modulus we have a spiral ganglion that includes cell bodies of sensory neurons. The dendrites go to the to the organ of corti or organ of hearing, and the axons form nerve which will carry information to the brain. Then here we are looking at the organ of corti structure. Now, before talking about it, we have to mention the walls of the spiral duct. As we know, we see here spiral duct has triangular shape. It has basal wall, basilar wall, then medial wall, which composed of Reisner's membrane or vestibular membrane, and it has lateral wall. Lateral wall is covered by vascular epithelium, which is called stria vascularis. There is the special cells that produce endolymph into this cavity of the cochlear duct, which is filled with the endolymph. So the endolymph is produced here. Then vestibular wall, it covered in both sides with simple epithelium, and between them interposed connective tissue lamina. Then in the basilar wall, we have this organ of corti or hearing. The histological structure of the organ of hearing, the cells which form it, we are looking here. There are actually there are mainly two kinds of cells here. The outer hair cells, there is the hair cells and supporting cells. All of them we can subdivide into two groups: inner group of cells and outer group of cells. And so accordingly we will have inner hair cells, outer hair cells, inner supporting and outer supporting cells. Between them we have triangular shaped canal, which is called tunnel, and in the wall of this tunnel we have pillar cells, which also belong to the supporting cells. Through the inner tunnel pass nerve fibers of the nerve fibers which come from the spiral ganglion. Spiral ganglion, as we know, is located inside of the modulus, and from here the dendrites of those neurons come to form synaptic connections with the hair cells. So the neurosensory cells here, also the hair cells, and which also subdivided into first type and second type. First type have rounded base, and other one, second type, have columnar mainly shape, cylindrical shape. Then on the surface of these cells we have stereocilia, and approximately in the inner cells we have 60 to 80 stereocilia, and in outer group of cells approximately 100. If in inner cells they have V-shaped organization, localization, and arrangement, in the case of the outer hair cell they form W-shaped ways. So these are the hair cells, and supporting cells are of several types. So we have here, as we mentioned, the pillar cells. Then we have phalangeal cells, which are located under the hair cells. Then we have other supporting cells, which are called according to the name of authors. So here we are looking at the modulus, which contains spiral ganglion here. The dendrites of these neurons which arise from this ganglion come to the organ of corti, and that is a cross section of one of the spiral canal, and that is scala media or cochlear duct, scala vestibuli, and scala tympani, which belong to the bony labyrinth. Then here these are the cells. Above the hair cells we have tectorial membrane. It is also gelatinous mass, and the stereocilia here embedded in this membrane. The changing of the position of the membrane leads to bending of stereocilia, and it leads to activation, excitation of these nerves, of these epithelial cells, and it leads to the information goes to the dendrites of the sensory neurons. By that way it is transmitted to the brain. Here we also can see the dictorial membrane, the sensory cells here, hair cells, then the supporting cells, the tunnel here, nerve fibers, and in this region, actually, that is the limbus region. That is here we are looking at the bony shelf. It is a spiral lamina, which is, pay attention to this region. It is a bony shelf which arises from the modulus. It is projected towards the lumen of the spiral canal, and this region is called limbus. And here periosteum of the spiral lamina is subdivided into two layers, two lips. There is the vestibular lip and the tympanic lip. Vestibular lip continue into the dictorial membrane, as we see here. That is vestibular lip that continues into the dictorial membrane, and that is tympanic lip that continues into the basilar membrane. And basilar membrane is membranous structure that contain collagen fibers that are going from the modulus towards the lateral wall of the bony labyrinth. Then here the first type of cells, second type of cells, then the phalangeal cells here. Why they called phalangeal? Because from the lateral part arise one like process which have shape of like phalanx digit, and it forms like support for the hair cells. Then that is a protuberance, stria vascularis, that is lateral wall of the cochlear duct. And here we have protuberance. It is a ridge of the bony wall that is covered by simple cuboidal cells. Then we are coming to here, that is a protuberance. And these cells continue into cells of Claudius, Bouchard, all of them, these are supporting cells. And so that is the organ of Corti. And now let's all talk about the mechanism of sound perception by the organ of hearing. As we know that sound waves is received by the external ear, and then the sound waves pass through the external auditory meatus and reach the tympanic membrane. And the tympanic membrane receives the sound waves, and it converts it into the mechanical energy that it will undergo vibration. And vibrational tympanic membrane set these ossicles of the middle ear into motion, and because of the lever activity and articulations, the force of the mechanical energy when it reaches the oval window where stapes is attached is increased by 22 times. As we know that it is the result of two activities. First of all, we know that the diameter of the tympanic membrane is 17 times greater than the diameter of the oval window. So it means the increase of the force of vibrations by the 17 times. And then because of the lever activity of the lever action of the joints, articulations of the bony ossicles, it will increase to 1.3 times. So 22 times the force of movement will be increased when it will reach the oval window. And the vibration of the in the oval window, it set the fluid, which is the perinymph in the vestibular scala vestibuli, into motion, because we know that oval window have connection with the scala vestibuli. So the motion of the fluid, the waves that is created in perilymph, it will go over the scala vestibuli and reach the helicotrema at the apex of the cochlea, where it will be transmitted to the scala tympani. And scala tympani, we know that at the terminal part is terminates at the round window, which is covered by just membrane. So because of the fluid is incompressible, the waves that is created in the perilymph will set the basilar membrane into motion. And as we know, on the basilar membrane we have organ of Corti, and also vibrations in the perilymph. It leads to vibration of the vestibular membrane, which is located over the that is medial wall of the scala media or spiral duct, and it also will set the fluid, the endolymph, into motion. And finally, we will have displacement of the tectorial membrane over the organ of Corti, and the stereocilia, because stereocilia are embedded inside of the tectorial membrane, and the position of this stereocilia will be changed. That will lead to opening of the ion channels here. There is mainly opening of the potassium ion channels, and entrance of the potassium ions into the hair cells will cause depolarization there. That depolarization will be transmitted via the nerve into the spiral ganglion and via cochlear nerve to the brain. Here, that is the crista ampullaris, which is organ of vestibulum or organ of balance. Here how we will sense the angular movements of the head. We are looking here at this organ, that is the neuroepithelial cells. As we know, on the apical surface there are also stereocilia, and besides it there is also kinocilia. Stereocilia, they, as in the organ of Corti, they are interconnected with each other via bridges. That's why they cannot move freely, and they may just bend at the basal part where these processes arise from the cell body. And the kinocilium is immovable process. During the changing of the position of the head, during movement of the head, changing position of the head, the cupula, which is located above this organ, will change its position, will bend, and bending of the cupula will lead to bending of the stereocilia. If the stereocilia, as we know, as we see here, they're located in some order, that the longest stereocilia located near to the kinocilium, and the shortest is far away. During bending of the stereocilia towards the kinocilium, the cells undergo excitation and nerve impulse is generated, that is transmitted via the nerve terminals to the vestibular ganglion and to the and via the nerve fibers to the brain. And when this stereocilia will move in opposite direction, far away from the kinocilium, the cell undergo inhibition. Here we are looking how that is the vestibular ganglion that collect nerve fibers from the organ of balance or equilibrium, and the spiral ganglion, which collect nerve fibers from that coming from the organ of Corti, and these two nerves will form the vestibulocochlear nerve that will go to the pons where located their subcortical centers. And finally, the organ of taste. Taste organ is located in the tongue. As we know, the dorsal surface of tongue is covered by the papilla. There are different types of papilla, vallate, circumvallate papilla. Here we are looking at the fungiform, filiform papilla. Exception filiform papillae, all of others foliate, vallate, fungiform papillas. On the lateral surfaces, they have taste buds. These are receptors for taste discrimination. If we will look at the structure of the taste bud, it has onion shape, and inside of it we have dark light cells, intermediate cells. All of them belong to the taste receptors. Also, we have basal cells, which are stem cells for replacement of all of other cells. On the apical surface, there are microvilli, and to the base parts come nerve fibers, which will carry these taste impulses, nerve impulses about the taste sensations to the central nervous system. That was the lecture on the sensory system. So see you on next lectures.